All right, good morning, everyone. So this morning, I will give two lectures, one on clustering, trajectory inference, and also geometric properties in the context of single-cell data. And the other is about how we handle sparsity, or how people in the literature have been developing methods to handle the sparsity in single-cell RNA-seq. So the first lecture likely is gonna go a little bit longer than 1 hour because of the additional material I added on top of what I had last year. But well, I'll try to go with a reasonable pace, but while still being able to cover all these material, hopefully. Okay, so let's get started. Okay, so the first topic for this lecture is a clustering analysis in the context of really single-cell RNA-seq. And I want to do that using the material published in, I would say, probably the first high throughput single-cell RNA-seq paper. So in general, clustering analysis starts with this gene cell count matrix, and it is intended to examine similarities among cells, among these columns, to identify groups of cells or groups of columns that are highly similar to each other, so that later, when we do dimension reduction to visualize the data, we may see these islands corresponding to clusters of highly similar cells. And each of these clusters may show some marker genes that are specific to those clusters that allow us to perform interpretation. Okay, so the paper that I'm going to use to discuss, the algorithm pipeline that I'm going to discuss is coming from this paper here. This is, again, in my opinion, the first single-cell RNA-seq paper that really showed the ability to generate such data in very high throughput. And people who wrote this paper, especially Rahul Sathija's group, later, he was in the Broad Institute when this paper was published, but later he moved to New York and starting his own lab. And he has been developing and maintaining this Saret R software package tool that can actually do a lot of things, including clustering, data integration, spatial inference, etc. So what I want to do today is I want to talk about their clustering pipeline. Okay, so as we've already actually mentioned earlier, that single-cell RNA-seq data is highly sparse. So in this particular data set, this is, again, one of the very first high-throughput single-cell RNA-seq data set. When we look at the gene cell count matrix, there were roughly 24,000 genes and around 40 to 50,000 cells. So when we look at this matrix, 97% of the entries in this matrix is 0. So, and if we look at, for example, number of genes detected per cell, for some cells, the number of genes detected can reach as high as 8,000, 9,000. But for the majority of them, the number of detected genes was below 1,000. This is, of course, an earlier data set. And if you look at the data set today generated by 10x Genomics, for example, then this percentage sparsity of zeros is, I think, still pretty much always above 90%, maybe in the 93, 94% kind of range. So it's better than before, but it's better than this initial data set, but not so much better. But of course, there can be an argument that maybe many of those zeros are supposed to be zeros. Right? And we're going to talk about that in the next lecture. All right, so again, this plot is just to show you the sparsity of the data. And now let's actually go through their data analysis pipeline. So what you're going to see in the next couple of slides is that I'm going to use sort of diagrams of boxes of sort of these squares or rectangles to represent input and output shape of each step of the analysis. Often when I talk to my own students, when they read a paper that presents a complex pipeline, I actually would want them to draw the diagram that I'm going to show you so that they will have an overall picture of kind of the input-output relationship along the process, even though they may not 100% understand the mathematical underpinning of each data processing step. But anyway, let me go through this first. So the starting point of this data set, as I mentioned, is this gene cell count matrix of 24,000 genes, almost 50,000 cells. And this data matrix is 97% sparse. So if we try to apply any clustering algorithm, off-the-shelf clustering algorithm to this data set, then for sure we're gonna fail miserably, because existing standard machine learning algorithms, none of them are really designed to handle data with such sparsity. So some pre-processing to improve the quality of the data is definitely needed. So their step number one is a normalization step, and this is called library size normalization. So this step is done column by column or cell by cell. So you take one column, right? This is the gene expression profile for one cell. You divide this column, every entry in this column, right? You divide it by the total sum of this column, and that's the total UMI count, and then multiply by a nominal number of 10,000. And then that's going to be the normalized version of this cell. And we do this for every cell independent, such that after this library size normalization, if we look at the sum of each column, the sum is always 10,000, okay? So this 10,000 is just a nominal number that people often use, but I've also seen other single-cell RNA-seq papers where they did their normalization to 100,000 or even 1,000,000. So this number, again, is sort of just a nominal number. And there's kind of nothing really magical about it, just to ensure the total amount of kind of normalized expression, right, for each cell is the same. Now, this normalization step, you can say that this is sort of fair, because there will be variability in terms of how much signal we capture from each cell, and we normalize that. While at the same time, one can also argue that this normalization may not be perfect, because among all the cells we captured here, among all these cells, some of them are bigger, some of them are smaller, and the bigger ones may naturally have more mRNA content and more sort of gene expression level compared to cells that are smaller. And this kind of normalization will remove that biologically meaningful variation, right? So this is a step that I almost always do, okay? But at the same time, I also would sort of think about when I do this, what kind of information I may lose, and do I care about those information? If yes, then maybe later I also want to go back to the unnormalized data and see whether the clusters that I obtain, they show, say, different total EMI count before normalization. All right, so the next step is log transformation, and the effect of this step is actually similar to one thing what Greg showed yesterday about the effect of log transformation on bulk RNA-seq. So if we take, say, all these numbers, we show the histogram of all these numbers, we'll see a exponential decay kind of distribution. There are many, many entries with 0 or 1 or very small values, and the entries that have large values in this matrix is very rare. So that kind of exponential decay. But after we take log transformation, okay, then we look at the transformed numbers, and we can draw another histogram, and we'll see that the distribution will now look a little bit more Gaussian-like. And this transformation, turning the numbers or distribution to be more Gaussian-like, will actually enable subsequent analysis to behave better, because a lot of the statistical tests and machine learning algorithms were actually developed with this Gaussian assumption in mind. Okay, all right, but well, this normalization and also this log x+1 transformation, they actually do not change the sparsity of the data. So at this point, the dimension of the data, process data, is the same as the original, and the percentage of sparsity in the data is still the same. Okay, so the next step, what the authors did was they chose to filter cells. Okay, so in this paper, what they did was they only kept cells with more than 900 genes detected. So again, this is the same plot I just showed. So for many cells, these cells, the number of detected genes is relatively small, right? And what they did was they say that we want to only keep cells that have sufficiently large number of genes detected, so that those cells likely will contain enough useful information for us to define cell types and cell clusters. So let's focus on those cells first. And the ones that are thrown away later, they will have a method to bring them back. But in terms of defining clusters, they want to use kind of more reliably profiled cells to define clusters. Now, if you look at analysis pipelines people do nowadays, this sort of cell filtering step is like almost always a QC step that people include. And sometimes there will be just this threshold for lower bound so that they people exclude cells that with too few genes detected. But sometimes people also apply an upper bound, saying that for cells that have too many genes detected. So for example, here we have a small number of cells that have almost 9,000 genes detected. So those cells, maybe we should also exclude them, because when I mean during the library preparation process, right? The RU encapsulation of individual cells, it's actually possible that one RU droplet will encapsulate 2 cells plus one bead. So then in that case, at the end of our bioinformatics pipeline data analysis, mRNA content for both cells, they will, because they're captured by the same bead, they will share the same cell barcode. So to us, it will appear to be just to be one cell, okay? But then this particular bead has access to mRNA content for 2 cells, and it's likely that this bead is able to capture more mRNA content and then enable more genes to be detected, right? So here for cells, or quote unquote cells, that have too many genes detected, it is possible that these quote unquote cells, they are doublet. Right? So sometimes when people filter cells, they may apply 2 threshold and say that only, say, cells within a certain range of genes detected, we're going to keep those and consider those as high quality cells to perform clustering and define cell types. Okay, so in this paper, they only had one threshold here, a lower bound threshold, and this threshold excluded roughly 75% of their 3/4 of their cells. Right, and then in their next step, they filtered genes. Okay, so the way they did it, is to compute the mean and dispersion of every gene. So the raw mean and dispersion shows sort of some kind of trend, but after detrending, so what they had was each dot is a gene, and the horizontal axis is the mean of the gene, the vertical axis is the dispersion of the gene. And they want to identify genes above a certain line here, above, say, a certain line here, this line, okay? So all these genes, okay? These genes are ones that have larger dispersion compared to other genes with similar expression level. And those genes, because they vary more than the other genes, those are considered as highly variable genes. And these highly variable genes, they are likely to contain more information about various cell types in the data. Okay, so imagine if you have one gene that's lowly variable, right? It's a housekeeping gene that's consistently highly expressed in every cell, then that gene is not useful, right? So, or if you have one gene that's expressed in only one of the cells, right? Or is consistently sort of very lowly expressed everywhere, then those genes also show low variability and not really useful for defining distinct cell types. So it is only the ones that are that have higher dispersion compared to the others are going to be more useful. So in this particular paper, what they did was they chose a threshold here, okay, and then only kept roughly 400 genes above the threshold. Okay, so, and now this is going to be the data that they use to define cell clusters. So this filtering step in this paper is super aggressive, right? We quantified 24,000 genes, and eventually we're only keeping 1% of the genes to perform clustering analysis and define cell types. Okay, so today, if you look at pipelines that people often use, this number is usually larger. People often choose to use top 2,000 highly variable genes instead of just a few hundred. But in this paper, 400 worked well, but usually people would include more genes here, somewhere around 2,000. Okay, now after all these filtering steps, we are kind of focusing our analysis on these highly variable genes, some of the more informative genes, and also the cells with larger number of genes detected, so higher quality cells. But even with such filtering, if we look at this matrix, the sparsity is still pretty high, right? And in their next step, what they did was they performed principal component analysis to further reduce this 400-dimensional data down to a 32-dimensional data set. Okay, so the reason why they chose 32 as the number of PCs they want to keep is actually the Jack-Straw procedure that we discussed yesterday. It was among the procedures we discussed yesterday, Jack-Straw was the only one that examined feature loadings to decide which principal components are informative and should be kept. Okay, so here on the right, what they showed was a scatter plot of PC1 versus PC2. Each dot is a cell. Okay, so here we see a clear pattern. It's not a hairball, right? It's not like a Gaussian distribution without any correlation structure, right? And if we look at PC3 versus 4 and so on and so forth, we see that only when we arrive at kind of the last two PCs they chose, we start to see that majority of the cells just form a hairball without correlation. But still, there are some sort of these protrusions, right, that seem to show some correlation structure. Now, if we go further, then we will see even less correlation structure, which is one way to say that the top PCs that Jack-Straw procedure decided to keep, majority of them are indeed showing kind of non-random variations. Okay, so after the data was reduced to 32-dimensional, okay, their next step is a t-SNE, is a further dimension reduction visualization, because a 32-dimensional data set in the PCA space, we still cannot see it, right? In order for us to really visualize the data so that we can show something on a 2D piece of paper, right? So they did further dimension reduction using the t-SNE approach we discussed yesterday. Okay, and here is the t-SNE plot where we can see a lot of these islands. Okay, so the reasons why we see this are twofold. One is that, well, among all these cells profiled, and by the way, these cells are harvested from mouse retina, and there indeed are a variety of cell types there with distinct gene expression profiles. So there are clusters. That's why t-SNE shows us clusters. And plus, there is also a second reason, as we discussed yesterday. t-SNE actually is an algorithm that's sort of designed to enhance cluster structures, so that t-SNE is also encouraging kind of clusters to appear in this visualization. Now, what they did next was, well, this t-SNE step was able to map, say, these 13,000 cells onto this 2D space. But remember, earlier on, we have a step to filter cells, and 3/4 of the cells were filtered away, right? So now, in this paper, one thing they did was they used a kind of a, I would say, t-SNE related trick to bring all the cells that were filtered away here and bring them back also into this 2-dimensional visualization space. Okay, so the t-SNE plot here has only 13,000 cells, but after their next step, they have a t-SNE plot that has all the 50,000 cells. Okay, and then their next step was density-based clustering. Okay, so they have this t-SNE map, right, containing many dots in it, and they form clusters or islands as we see. And if we perform density-based clustering analysis, the algorithm will essentially identify each island as a particular cluster. Okay, so here density-based method works well is because, well, the data that goes into density-based clustering is already in 2-dimensional. Okay, what we discussed yesterday. Density-based clustering usually will work well when the input data is in low-dimensional space. Okay, and once they've performed density-based clustering, they saw 49 clusters here. They're like maybe 49 islands if we count, right? And later, what they did was they did pairwise differential expression analysis to identify how many genes are differentially expressed between each pair of these 49 clusters. And they noticed that among these 49 clusters, certain pairs have very, very few differentially expressed genes, so that they, the authors did not feel that comfortable to call them as distinct cell types, and they decided to merge them. So there was this heuristic merging step based on differentially expressed genes, and eventually they ended up with 39 clusters, and those 39 clusters are what they used for further biological interpretation and experimental validation. At the end of the day, they presented this t-SNE plot with 39 clusters, and they show that different clusters show distinct marker genes with expression consistent with the what they expect. They also showed that among these 39 clusters, there were some, at least one, maybe more, cluster correspond to cell types that are not known before, and they had some experimental validation to validate those novel cell types. Okay, so this is their whole data analysis pipeline. Okay. So here is a kind of a text version describing their pipeline. So where I want to draw your attention to is how they did clustering. So their initial approach published in this paper was they have the PCA space, right? They first perform PCA to reduce their data dimension to 30, and then they did t-SNE to reduce the data dimension to 2, and then clustering was based on this 2-dimensional space. So essentially, and in the 2-dimensional space, because the data already look quite sort of cluster-like with all these islands, so essentially, density-based clustering, clustering is almost fully defined by the t-SNE dimension reduction step. And density-based clustering is just sort of a convenience for them to read out how many islands there are in an automated way. But later, okay, in the later pipelines that the SISG group suggested, they changed their implementation. So instead of performing density-based clustering on the t-SNE space, what they did was alternatively they chose to use community finding algorithm applied to the PCA space to define clusters. Okay. So the whole procedure is going to be that we start with raw data, we perform some normalization, library size and log, and then we filter genes, we filter genes and filter cells, so that we focus on the higher quality part of the data, and then PCA reduce the dimensionality, and then community finding algorithm to form cluster. Okay, and in parallel, people would use either t-SNE or UMAP to generate a visualization, but the visualization generated and the clusters defined generating the 2D visualization and defining the cell clusters, these two are done in parallel. One by UMAP/t-SNE and the other done by community finding. And later, the clustering results are projected onto the UMAP or t-SNE. Usually they agree with each other really well, but sometimes there are disagreements. And nowadays, I think the common consensus is that it is better to define clusters based on the PCA space, so that because otherwise, if we do it the previous way, then we're 100% trusting the UMAP or t-SNE visualization to define clusters. But that dimension reduction step, because it's compacting information into just 2D, and there could be kind of undesirable loss of information or even artifacts introduced. So that I think later that's why people later moved away from defining clusters based on the 2D visualization space, and instead try to define clusters based on the PCA space. Okay, so now here is sort of the whole pipeline again. And as I commented earlier, when I talk to my own student about reading papers, with complex pipelines, I always ask them to draw such a pipeline so that we know what is the dimensionality of the input and output of every step. and then we'll have a big picture of how the whole kind of workflow goes. And after that, we can try to spend more time to understand, for example, what library size normalization does, and how does PCA work, and how does, for example, clustering work, right? So, and this is kind of all I want to say about kind of the existing standard clustering pipeline that people often use. Okay. All right, so I'll take a short pause here. There is one question about how do you decide a merging of clusters? So what this paper did was, this paper looked at a number of differentially expressed strings between each pair of clusters and say that if the number of differentially expressed strings is fewer than 100, they say that there isn't enough distinction between these two clusters, and they decide to merge it, right? But 100 is also, well, it could be a large number. You can certainly use other thresholds. Well, at the same time, you know, the number of differentially expressed strings is also somewhat affected by, in total, how many cells you have, right? So in this study, they have roughly 50,000 cells, okay? And with this many cells, let's say all cells are equally distributed in each cluster. So each cluster has a good number of cells, right? And a larger number of cells per cluster will actually enable you to see kind of more significance, because you have more power. And power and sample size, they always kind of are somewhat positively correlated. Now, as opposed to, you know, if the study only generated 5,000 cells, okay, then for when they do differential expression analysis of their clusters, they will naturally see much fewer number of differentially expressed strings. So so in that case, maybe, you know, 10 would be a good number, right? 10 would be a good threshold to say, you know, if two clusters have fewer than 10 differentially expressed genes, we say they're the same cluster. Okay. So and that means, you know, in your own practice, right, when you try to heuristically determine whether or not two clusters should be merged or they represent distinct biology, what would be the appropriate threshold for number of differentially expressed genes you want to require? There really isn't a universal answer. It's somewhat related to the total number of cells you have. Okay, so another question from Justice. Yes, I do use MATLAB a whole lot in my demonstration, but in fact, MATLAB's bioinformatics toolbox is not sufficient to carry out single-cell RNA-seq analysis. MATLAB compared to R, it always lags behind. And in my own research, I actually write all the packages myself. So I think for single-cell RNA-seq, R and Python would be the platform that can provide a lot of packages and help if you are happy with using existing tools out there. In my own research, what I do is I want to develop my own tools. So that's why I have been feeling okay to work within MATLAB. But if, say, I have time to sort of totally revamp my own programming, rewire my own programming sort of mindset, I probably will switch to Python. Okay. All right. So okay, so there is one more question about how the cluster would differ if they did not bring back the removed cells. My gut feeling is that the clustering results will be highly similar. Okay. All right, so now let me move to the next topic about trajectory inference. So as we discussed yesterday, trajectory inference will be a meaningful analysis if the single-cell RNA-seq data is generated from a induced differentiation kind of experiment, or developmental biology kind of experiment, or drug response kind of experiment. because in these experiments, we expect to see cells from, say, an unperturbed initial state, and then gradual transition through intermediate checkpoints to some, say, destination cell state. And because we expect to see cells along this entire biological process in high-dimensional space, the data may form some kind of trajectory waiting for us to extract from the data. Right? And one algorithm, one of my favorite trajectory finding algorithm is called diffusion pseudotime. Okay? Well, partly because it is relatively easy to explain, but there also are many other reasons why I liked it. But let's go through this paper first. Okay, so similar to the clustering pipeline, I also want to use these boxes and diagrams to describe this DPT analysis algorithm. So the starting point is view the gene cell count matrix. Okay, 20, 30,000 genes, however many cells we may have in the data. So as first step, what DPT does is it computes this pairwise distance matrix, which is just Euclidean distance between two columns here. Okay, so now this pairwise distance matrix, the dimensionality of it is number of cells by number of cells. It's a square matrix. Right? And of course, I mean, before doing this, they also have pre-processing steps of library size normalization, log transformation, and sometimes they also do cell cycle normalization to remove cell cycle effects in the data. Now, after they have this pairwise Euclidean distance, okay, well, the authors know that especially when we're interested in finding trajectories, still it is the short distances that matter more than long distances. So what they did was they defined something they call affinity, okay, which is of the same dimensionality, is still cell by cell, right? Number of cells by number of cells. And in this affinity matrix, each element is defined this way here, okay? It's defined as, again, E in exponentiation, and then we have negative sign, and then square of the distance, okay? So this is pretty much the same as the mathematical or the probability formulation in t-SNE and UMAP. It's trying to heavily discount large distances, while small distances will still receive some non-trivial value, okay? So now if we compare D and K, okay, we'll see that large elements in the D matrix will become close to 0 in the K matrix. Small elements in D matrix will still have non-trivial value in K. Okay. And after that, what they did was a step they call normalization by density. So from K to W, W is of the same size as K. Okay, so each element of W is computed by, you take the corresponding elements in the K matrix, and then you divide by the column sum and the row sum corresponding to that element. So this is just a normalization step. Okay, and after that, this paper did another normalization from W to T tilde, right? And the formula is actually almost the same, right? From W to T, the normalization is still done entry by entry. So for each entry in the T tilde matrix, they just look at the corresponding entry in W and then divide by the product of the row sum and the column sum with a square root here. So this formula, okay, compared to the previous formula, these two formulas are pretty much the same. And in fact, I still don't really understand why they needed two steps of normalization, because again, they're almost the same. But anyway, it is a component that the authors decided to keep in their pipeline. In my mind, I would consider this T tilde and this W matrix to be the same. Okay, and then after that, what they did was they compute what they call diffusion. Okay, and the whole idea here is that when they generate this D matrix, when they turn this D matrix into this affinity matrix, again, if we look at one particular row, okay, we'll see that all the large values are zeros, close to zeros. All the small values are kind of non-zeros. And then they use this Markov transition kind of idea to interpret this affinity matrix. They will say that if we are, let's say, sitting in, if we are looking at one cell, okay, and we want to jump to other cells with whatever probability distributions, then the likelihood of us jumping from this cell to a nearby cell should be non-trivial, should be non-zero, whereas the likelihood for us jumping from, say, where we are to another cell that's far away, that likelihood should be very, very close to zero, okay? So this affinity matrix K here, each row, you can think of it as the likelihood or probability of going from one cell to other cells, and we only have high likelihood to go to nearby neighbors. And W and T, these are just normalized versions of K, so their interpretations are the same. Okay, so here this T tilde also is a probability transition matrix. And then in sort of Markov/graph theory, so if you have a matrix that represents a one-step transition. So let's say from this cell, if I'm only allowed to jump one step, where can I go to? I can only go to my nearest neighbors, right? But if you compute T tilde square, you get another matrix. And this second matrix actually would represent a two-step Markov transition probability, okay? So if you look at T tilde square, that matrix will tell you if you're at one cell, okay, and you're required to make two jumps, where can you go to, okay? And so the T tilde square matrix will be less sparse because with two steps, we can go to our neighbors and their neighbors. We can go further away, right? And we can also do, say, T to the third power, T to the fourth power, and T to the infinity power. And that's basically how this M matrix is computed. So let's just ignore this phi here. Okay, so this M matrix is just T plus T squared plus T cubed plus T to the fourth power, etc., etc. And this infinite series actually has a nice mathematical formula that we can use to easily compute. Okay, so this M matrix here represents the possible transition among cells if we consider all possible number of steps. One step, two step, three step, because we're summing up T squared, and T cubed, etc. Okay, so this is the overall transition probability. And their very last step was that now for two columns, if we look at two columns or two rows, well, here, let's see, well, two rows. This matrix is symmetric, so it doesn't really matter column or rows. But if we look at two rows, okay, one row represents the probability distribution of where we can go to if we start from this cell. And another row represents, again, the probability distribution of where we can go to if we start from another cell, right? So if these two rows, if this row and this row, if these two rows are highly, highly similar to each other, okay, meaning that regardless of whether we start from here or here, where we can go to is highly similar with a similar probability distribution, then it indicates that this cell and this cell, they must be at pretty much the same location. All right. So the probability jumping profile for two cells, okay, that's this thing and this thing here, their difference is defined as the diffusion pseudo time between two cells or the distance between two cells, okay? So this is sort of a quite kind of winded road to get to this diffusion pseudo time matrix that represents the distance between two cells. And because of how they discounted large distances, so this diffusion pseudo time matrix actually can roughly approximate the geodesic distance in isomap, and also the kind of the k-nearest neighbor graph and the geodesic distance concepts in UMAP. Okay, so now if I try to arrive at the same place, but use an alternate pipeline, so one thing I actually can do is that we can follow the UMAP procedure. So starting from the raw data, okay, let's generate a k-nearest neighbor graph first, okay, and then on the k-nearest neighbor graph, we use the UMAP formula or this formula here to give weights on each edge. Okay, so now we have a graph connecting many cells, and each edge has some weight. And then for every pair of cells, I can try to find the shortest path connecting these two cells on the graph and use the total weights on that path to define the distance between two cells. So if I do it that way, I will also be able to get this something that's very similar to this DPT matrix. But the difference is that all these calculations, these are pretty much just matrix operations per row, per column. So they actually do not really take much time. But if I generate a k-nearest neighbor graph containing thousands and thousands of cells, and I try to find shortest path on that graph, which will be a computationally very, very expensive task if I want to do that for each pair of cells. Okay, so to me, I think the key advantage of kind of this pipeline getting to this point is that they are able to, well, this DPT matrix is able to approximate geodesic distance using a series of fairly inexpensive computational steps. Okay, but overall, you know, you can interpret this DPT matrix as geodesic distance on k-nearest neighbor graph. All right, so once they have this geodesic distance on the k-nearest neighbor graph, or I mean, geodesic distance in between each pair of cells, then they have a series of more kind of computer science operations to identify within this data set, right? How many branches there are, and which cells belong to which branch, and how are cells ordered along each branch. Because well, at the end of the day, we want to extract a trajectory out of this data, and we want to know, is this trajectory just a linear line, or does it have any branching points? And also, how are cells ordered along this progression, this trajectory? So the way they do it is that first of all, they say, given this DPT matrix, right, they randomly pick one cell, or, well, yes, once each cell is unknown, right? So they randomly pick one cell, and then they try to find the cell that is most far away from the randomly picked cell, right? And distances are defined in this DPT matrix. So with this pairwise, this DPT matrix, they know the distance between each pair of cells, right? So for any given cell, which cell is the most far away from it is easy to find. So if this is what we pick, then the cell that is most far away from it is this one, right? And then we go from here and ask, what is the cell that's farthest away from this this one? Then likely will be either here or here, right? Let's say we get to this distal point. And then they find a third cell, which is the one that is the most far away from R1, R2, and the third cell must be this. If we are looking at a graph structure that has 3 arms. If we are looking at a graph structure that has 4 arms, maybe there's a small arm here. And if this arm is really short, then the distal end of this arm will likely not win the process when R1, R2, R3 are selected. So this is a simple procedure to essentially identify the distal point of the 3 largest branches in this graph. Okay, and after that, what we do is that what they do is that they compute the diffusion pseudo time from all data points or cells from each of the distal end. So from R1, okay, the diffusion pseudo time for this node is, or distance, is going to be 2, and then the next one is going to be larger, etc., etc. But then when we go to these 2 branches, then we'll say that the diffusion pseudo time from this data point to R1 and the diffusion pseudo time from this data point to R1 is going to be similar. Okay, maybe one is 9, the other is 10, or maybe this is 9.2 or something like that, right? So the diffusion pseudo time along these two branches with respect to the starting point R1, both of them will gradually increase, but they share overlapping values or overlapping range. Okay, and we can do the same. So starting from distal end R2, we can label every cell according to how far away they are from this distal end, and we do the same for R3. Okay, So once they label cells in these 3 different ways, according to the 3 distal ends of the major arms, then their next step is to identify where these branches would, where these 3 different branches would kind of meet and converge, right? So what they do is that they say, oops, what did I do? All right, just one moment. My PowerPoint just restarted. Close. And no, this is not the right one. All right, so we were here. All right. Okay, so what they do was that they say, let's start from distal end R1 and gradually walk down the path. So if we look at the first 4 nodes, okay, then we can look at, for the same set of 4 nodes, the values we attached to these nodes with respect to R2 and R3, are they highly correlated? If we look at these numbers, okay, they're highly correlated. Okay, so that's great. That means this correlation is 1. But if we look at the rest of the numbers, or the numbers associated to the rest of the nodes, okay, are they perfectly negatively correlated? Not necessarily, right? So for these nodes, okay, for these nodes that do not belong to the R1 branch, okay, so from here all the way to this point is increasing, but from here all the way to, again, the other distal end is monotonically decreasing. So now for these nodes, okay, the numbers of the DPT distance we annotated with respect to R2 and R3, they're perfectly negatively correlated. But since we are also we are looking at only sort of the rest here, right? So this correlation is not really -1. But if we say gradually walk down the list, walk down the R1 branch up until when we reach the kind of the convergence or the branching point among the branches, then what we'll see is that for the cells that we hypothesize to be on R1 branch, the numbers we attach to these cells are perfectly positively correlated, and the numbers we attach to the nodes corresponding to the rest of the data, they're perfectly negatively correlated. So now we have a +1 minus a -1, and that maximizes sort of this statistic they look at. Okay, so what they do is essentially they go from, say, the node R1, gradually walk down the list of cells that are further and further and further away from this distal end, and they monitor this quantity. Okay, and when this quantity reaches maximum, they know that they have arrived at the branching point that defines the end or the starting point of this branch. Okay, and they also do this for all the other branches to know, for example, the R2 branch starts from this node and it goes all the way to here and then it ends. Okay, now after this exercise for each branch separately, they would know which cells belong to which branch, right? And then what they can do next is that, okay, so first they repeat this for each starting node, so that they, again, for each of the 3 branch, they know which cell belong to which branch. And after that, they can actually repeat the entire process. And they say, now that I know which cells belong to branch number 1, okay, then let's take branch number 1 cells and redo all the analysis and see whether branch number 1 can be further divided into 3 sub-branches. And they can do that for branch number 2, branch number 3 separately. And this allows them to really extract very complex tree structures. But of course, in reality, the data people often show very often only have 3 branches because people only run this once. And also, if you just try to run it iteratively, then you may be able to get, you may be getting too many branches that are very, very difficult to interpret. So here is actually the data presented in the DPT paper. So they have a visualization, they call it diffusion diffusion map that encourages the visualization to show trajectories. And here we see a trajectory that has 1, 2, and 3 major branches. And after they order cells along these branches, they can show a heat map like this, where say all these cells, each column is a cell, and all these cells, they are ordered along one of the 3 branches. And then these are cells on the 2nd branch, these are the cells on the 3rd branch. And when these cells are ordered, they start to see that, hey, along these ordering, there are certain genes that turn on very early on in the process, and there are genes that turn on later and later. And this helped them to identify really very detailed information of how different genes coordinate. They go up or down along, in kind of at different stages of this underlying trajectory or this underlying biological process. Okay, so here the ordering. Okay, so the assignment of cells to branches and also ordering of the cells along each branch is the output of diffusion pseudotime analysis. And other trajectory finding analysis also have kind of similar output structure. All right, so here just again a quick recap of their kind of data processing pipeline, which also can be represented using these kind of boxes with dimensions and arrows representing the analysis steps they do. And once they get to the geodesic distance in between each pair of cells, the process to extract a graph, extract the trajectory is kind of more computer science operations that cannot be represented using kind of data science operations. But anyway, so this is kind of my way of summarizing this particular paper. All right, so now let me actually quickly jump to the next topic, the geometric property of data. I think it's going to take me another 10 minutes to do it. So let's just do it now. So as mentioned actually yesterday, there are many algorithms that have been developed for clustering analysis and trajectory inference. So here are some screenshots from benchmarking papers that compared clustering and trajectory algorithms. Right, no need to read into the details, just give you an impression of how many algorithms that exist out there, right, before these benchmarking papers were written. And there certainly is a question of, say, if I want to do cluster analysis, which clustering algorithm I should choose? If I want to do trajectory finding analysis, which trajectory finding algorithm I should use? But actually, if we take one step back, there also is a question of, given one data set, if I do not have any prior biology knowledge of how this data set was generated, then should I do clustering, should I apply clustering analysis to this data, or should I apply trajectory finding analysis to this data? Okay, and the reason is because, I mean, these two analysis algorithms, each of them, they actually have its own bias. They have different assumptions, and they are kind of trying to morph and deform the data according to their assumptions. Okay, so for example, here are 3 different single-cell RNA-seq data sets. And on the left column, what we did was we followed the Surratt clustering pipeline to analyze these data. And we see that in all 3 cases, there seem to be distinct clusters, right? And the right column is the same 3 data sets, and we applied diffusion pseudotime and get the diffusion map of these data sets. And now if we look at the figures on the right, our interpretation is that these three data sets, all of them, they are trajectory-like, right? But then, once we see both, these two different type of analysis give us very different interpretation, then which one should we trust? Right? So actually, I have seen multiple published papers that kind of show this. Same data, they do trajectory analysis, they interpret the results, they do cluster analysis, they also interpret the result. And which, well, may be biologically meaningful, but can also be problematic. I don't really have a very good way to judge and say, that's the right thing to do or that's the wrong thing to do. But anyway, the same data with different algorithm, we see very different interpretation. Then that actually mean that potentially one of them might be somehow more appropriate than the other one, right? And that observation actually motivated my own research group to try to develop quantitative measure of the geometric property of the data so that we can try to quantify the clusterness and trajectoriness of a single cell data set, right? So if, and the way we did it was that we simulated many data sets, okay, with different geometry. So we simulated 3,000 data sets that all look like this with clear cluster structures. We also simulate data sets, many, with these noisy cluster structures. So it's still cluster-like, but it's not as clear. And we also have simulated data corresponding to clear trajectory and noisy trajectories. So now we have a large library of data sets, which we roughly know how they look like. And then we can use that data set as our test bed to develop scores that can distinguish clusterness versus trajectoriness. Or, I mean, scores that can distinguish these data sets versus these data sets. So one score we developed was entropy of distribution of pairwise distance, because if we compute distance between each pair of cells, if the data is cluster-like, then these distances are either short if it's a within cluster distance, or it's long if it's a distance between two cells belonging to different clusters, right? So, but if we're looking at a trajectory-like data set, then pairwise distances will have a more uniform or Gaussian-like distribution. And the entropy of this is higher than this. And that entropy is able to help us to differentiate cluster versus trajectory. So here are kind of distribution of scores we have for the 4 types of simulated data. And we can see that the trajectory-like data, they all get high scores. The cluster-like data, they all get lower scores, although some of them get high scores, right? So this score has some ability to differentiate cluster versus trajectory, but it's not perfect. But that's okay. We have other metrics that we've developed. For example, in topological analysis in math, there is this concept that's called persistent homology, which is quantified by, growing growing kind of a size of neighborhood radius from each data point, and then identify, by what time some of these merges would happen. And if data is cluster-like, the merging pattern is sort of one way. If the data is trajectory-like, the merging pattern is different. Okay, here we show the merging pattern of a cluster-like data set, where merges will quickly happen at the beginning, and then there's a big gap, and then one merge would happen when the neighborhood size, right, the radius grows to be similar to the distance in between distinct clusters. Okay? And entropy of this merging profile across these 4 different types of data set also is able to somewhat distinguish cluster and trajectory-like data set, right? If we put a threshold here, then we'll do a decent job in classifying left and right. Still, it's not perfect, but this is, well, a useful metric. We also have a metric we call magnitude, vector magnitude, and that's because, and this is designed by over-clustering the data so that we have many clusters, and then we jump across these cluster centers. Okay. Now, if we're in a trajectory-like setting, then from one cluster, we keep jumping to other clusters until we cannot find a nearby cluster that's not visited before. Okay. In a trajectory-like data, we can go really far. But in a cluster-like data, when we overcluster and we jump across the clusters, we're just sort of hovering around the same place and unable to go far, right? So magnitude will be small. And as shown here, this metric also has good ability to distinguish clusterness and trajectoriness. So we have two other metrics that I would like to skip the details, but just to show you on the right, the ability of these other metrics in terms of to differentiate clusterness versus trajectoriness. Okay. So at this point, we have 5 different scores we can compute for each data set. Okay, and we know that these 5 different scores, each of them have some ability to distinguish clusterness versus trajectoriness. Then what we did was we took this 12,000 data sets we simulated, and then we give each data set 5 scores. Okay, and now let's just imagine each data set is a point in a 5-dimensional space, right? And then we use UMAP to visualize that 5-dimensional space in 2D, and then this is what we get. Okay, we see that here each dot is a data set. Okay, we see that all the trajectory-like data sets are here. Okay, and then this tunnel here, or this bridge, and then everything on the right are cluster-like data sets. Okay, so now we're saying in our simulated data sets of all these different scenarios, if we examine these data sets using these 5 scores, we can separate the cluster-like data sets and trajectory-like data sets. Right? On the right is the same visualization, except that we just replace each dot by the actual scatter plot of the data. You see trajectories here, noisy trajectories here, and then you start to see clusters here and here. Okay. And now once we have this, what we can do is that we can use this UMAP as what we call geometric landscape to evaluate any real single-cell RNA-seq data set. So for example, this is a real single-cell RNA-seq data set. If we compute those 5 scores for this data set, and then see where this data set would be in our landscape, then this data set will be here in the trajectory region. And for this data set, if we do clustering analysis, we see trajectory. If we do trajectory analysis, we see trajectory, right? So now all these different algorithms and our geometric evaluation, they agree with each other. Here is another example of the same kind. Okay, so 2 more examples, data sets. So this data set, if we do the 5 scores, it will land here in the noisy cluster region. So in our geometric interpretation, we think this data set is cluster-like. And clustering algorithm shows clusters, trajectory algorithm shows clusters. So these are also agreements, right? And these are great. But then how about the cases I showed you initially, right? So these 2 data sets. Cluster analysis gave us clusters, trajectory analysis gave us trajectories. Now, according to our geometric evaluation, we think these 2 data sets both are trajectory-like. And looking at the biology behind how these data were generated, these both are developmental biology kind of setting. So we feel that it is probably likely that these data sets are indeed trajectory-like data. So 2 more examples where clustering analysis and trajectory analysis show some kind of disagreement. But for these 2 data sets, our geometric interpretation placed them in the cluster region. And now these 2 data sets, this one is a data set generated from distinct organs. And this data set is generated from flow sorted subtypes of T cells. Again, because of their experimental design, we feel that these data sets are likely more cluster-like, hence agreeing with our interpretation. So at this moment, actually a student in my lab is trying to apply this analysis to a collection of 200 data sets, so that we want to see sort of in a broader scale, when we try more data sets, do we still feel confident about our way of thinking about the geometric property of the data. So hopefully later this year, this paper will be published somewhere and will be available to everyone. Okay, so take-home message is that while we've talked about a clustering algorithm and a trajectory inference algorithm, and kind of how they work. And we see that choice of analysis algorithm actually can affect the interpretation of the data. So in data sets where we know the biology and we know how the data is generated, then that prior knowledge and our intuition probably will be sufficient to guide us on whether we should do clustering or trajectory finding. But then in data sets where we do not know what to expect, then we want to be careful with choice of the approach. We can try both and try to interpret both, but then in my opinion, I think we should try to form our own kind of opinion or bias about whether clustering or trajectory analysis is more appropriate for a certain data set. And also toward the end, I was showing these 5 different metrics that it is indeed possible to computationally quantify the geometry of the data, which can serve as another way to guide the selection of whether clustering or trajectory finding is more appropriate. And again, hopefully this paper will be published before the end of this year. Okay, so I will just end here and see whether there are questions. How do we Justice, I see your question about instances where x equal to y, but can you maybe elaborate a little bit on what you mean by that question?
Hello, can you hear me?
Yes, I can hear you now.
Yes, the DPT XY as the norm of mx colon minus my colon as a Euclidean space. And I'm asking if x equals y, how do you interpret that solution? Exactly this one, yes.
Okay,
if
so
x and y is the same, what happens?
So in the DPT definition, x is index of a cell, y is index of another cell, right? So when x equal to y, you're just looking at the distance between one cell versus itself. And of course, that DPT distance should be 0. Okay, so here, maybe I should just change all the xy's to I and j's so that it better represent that x and y, I mean, they are not continuous value variables. These are just indices.
Oh, okay, that's fine. Yeah.
All right, so there is another question from Emil. Should not clusters of data sets be tested? Okay, all right. So very good question. This is actually also something that we're thinking about for our next work, follow-up work for that geometry interpretation. So it's actually possible that we have a data set that's cluster-like, but then if we look at one particular cluster, this cluster is not a Gaussian ball. This cluster actually has some trajectory-like shape. Right? And therefore, it is actually possible to think about, say, for trajectory-like data, it's just trajectory-like. But for cluster-like data, each component or each cluster may be trajectory-like, or it may be just a Gaussian noise kind of a ball shape geometry. So that's definitely a follow-up we would like to do after we sort of finish the current work. A very good question. Okay, so let me stop sharing and stop the recording.